Welcome, everyone. My name is Rachel Massey, and I'm the Senior Science and Policy Advisor with CHE, the Collaborative for Health and Environment. We invite you to drop a note in the chat introducing yourself and where you're zooming in from if you'd like to. I'm going to start with a few bits of housekeeping before handing things over to today's moderator. CHE is a member group of the EDC Strategies Partnership, which is bringing you today's webinar. Partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEES, the Biomonitoring Resource Center, and the Health and Environment Alliance, or HEAL. CHE also hosts science discussion listservs and supports other collaborative programs focused on promoting environmental health and justice. For those of you who are joining by phone, we've posted today's slides on our website. You can download the slides by going to today's webinar page. You'll find the link in the upcoming webinars section of our homepage at healthandenvironment.org, then click on the slides and resources section of that page. And for those who are online, we'll drop the web page and the link in the chat now. We'll disable the chat once we get started so everyone can focus on the presentation, but please do type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentation. Also, if you'd like to activate closed captions, you can do that by clicking the button on the bottom of your screen. We are scheduled to last for 30 minutes, though we may go a bit over depending on the Q&A. And this webinar is being recorded for our archive. With that, I'll hand it over to today's moderator, Cheryl Patton, Director of the Biomonitoring Resource Center. Cheryl?
Thank you very much, and thank you everyone for joining this webinar on a really important study. I'm really pleased to introduce our presenter, Dr. Anna Young. She's a research associate in the Department of Environmental Health at the Harvard Chan School of Public Health and associate director of the Harvard Healthy Buildings Program. She earned her PhD and MS in environmental health from the Harvard Chan School and holds a BA in computer science and environmental studies for Yale University. Dr. Young will present a new study which used silicone wristbands worn by 243 office workers in the United States, the UK, China, and India as a method to collect exposures to chemical mixtures in the office environment. The research team extracted chemical mixtures from these wristbands and then examined these mixtures in human hormone receptor cell assays for total hormonal activity. They found that these chemical mixtures disrupted estrogen, androgen, and thyroid hormone receptors. It's important to note that these findings would not necessarily be obtained by studying single chemical exposures. The importance of mixtures needs to be studied more, and Dr. Anna Young has done a great job doing this. So, Anna, do you want to go ahead and begin your slideshow?
Hi, yes, thank you, Cheryl, Rachel, and Kristen for inviting me to do this webinar today. So I'm excited to talk to you more about this study that we just published a couple weeks ago that's about these hormonal activities of complex chemical mixtures in silicone wristband samples. So to put this into some context first, in the last decade alone, about 70,000 different chemicals were registered globally. And a fair amount of those aren't even publicly identifiable due to confidential business information. So we're being exposed to complex mixtures, including many potentially unknown chemicals. And many chemicals can be hormone disrupting or endocrine disrupting, which means they mimic hormones enough that they can disrupt certain health outcomes like fertility, pregnancy outcomes, development, thyroid function, diabetes, obesity, or even certain hormone-related cancers. And this is concerning because we know that hormone disrupting chemicals have often been used in all sorts of building materials, furniture, carpet and flooring, electronics, consumer products, and cosmetics or other personal care products. Now, one reason it's been so difficult to reduce our exposure to hormone disrupting chemicals is this phenomenon of chemical whack-a-mole, where we find out that one toxic chemical is harmful to health, we may eventually phase it out of production, only for other similar chemicals to just be swapped in as replacements that we later find out are also concerning for health and the environment. So it becomes this endless goose chase of trying to figure out what chemicals are being used next. And we've seen this happen time and time again with flame retardants, plasticizers, pesticides, PFAS forever chemicals, and all sorts of chemicals. And so the fact remains that traditional targeted research methods that test only one known chemical at a time can't keep up with the rate of new chemicals entering the market. So the research challenge becomes the need to evaluate our exposures to chemicals as the complex mixtures that they are in the real world, not only as individual chemicals. So that's why an overarching research question for our study was, how hormonally toxic are the complex chemical mixtures that we're exposed to in buildings? So specifically, we wanted to evaluate the hormone-disrupting potential of complex mixtures of both known chemicals and unknown chemicals that humans are personally exposed to in the real world. I've highlighted here some of the novel methods that we used to accomplish this, and I'll walk through each of those. So to get at personal exposures, we had participants wear silicone wristbands as passive samplers that while you're wearing them, they're collecting chemicals from the air and dust and products you're interacting with. They're useful because they're really simple, non-invasive. They don't have to be frozen immediately, so you can ship them around the world and sample remotely. What was really important to us is that they pinpoint external exposures and what you're exposed to from the environment you're surrounded by without including what you're eating or drinking. So in that way, they can allow you to control where and when you're sampling people's exposures. In our case, we had the office workers wear the wristbands only during work hours for 4 consecutive work shifts. So they stored them wrapped tightly in foil and bags overnight between work shifts. We recruited these 240 participants from 36 office buildings and cities in the US, UK, India, and China from the larger parent global buildings study of my advisor, Dr. Joseph Allen. And then we sent these wristbands back to our collaborators at Duke, Heather Stapleton and Nick Hergert, and they measured the wristbands for levels of 100 targeted chemicals, including flame retardants, plasticizers, pesticides, and some others. And then they also did something novel, which was to do suspect screening of other potentially unknown chemical features in the wristbands and to try to assign tentative identities to those features. And then finally, we sent other slices of the wristbands to the Biodetection Systems Lab in the Netherlands, and they conducted cell assays of the wristband extracts, where basically they exposed human cells in the lab to the extracts of the chemical mixtures on the wristbands and measured the amount of interference with estrogen receptor, androgen receptor, and thyroid hormone receptor. And I think this method is really cool and fun because they put a firefly gene into the human cell line so that it lights up in proportion to how much hormonal activity is happening. And you can measure that light in the lab. So these assays were helpful in this context because they're a way to rapidly quantify an immediate health indicator of exposures in a building. They also, unlike traditional targeted analyses, reflect impacts from all the chemicals in the sample, not just the ones that we know about or that we can measure in the lab. And then finally, they capture any combined mixture effects. For example, if one chemical has a worse effect when in the presence of another chemical. So briefly, the way this works in human cells is that chemicals can be agonists or antagonists. So normally, naturally in the body, a hormone like estrogen will bind to its hormone receptor, which activates and produces some important function. When a chemical is an agonist, that means it can look similar enough to the natural hormone that it can also bind and activate that hormone receptor. So then you have the hormones and chemicals going around activating the receptors, and that can be too much activity. On the other hand, some chemicals can be antagonists, which means they look similar enough to the hormone that they can bind that receptor, but they don't activate it. So they basically serve to just block off that receptor from being used, and that can lead to too much activity. So we're concerned about these because these nuclear hormone receptors regulate genes that are important for reproductive health, menstrual cycles, ovulation, sperm morphology, sperm concentration, and in the context of thyroid hormone receptor, growth, development, metabolism, and even brain function. So the reason this was novel is that cell assays have traditionally been used to test the toxicity of individual chemicals in isolation, or more recently to test toxicity of environmental mixtures like dust, which we've done in a previous study. But we wanted to take this one step further and apply it to people's personal exposures in the real world. So getting closer towards the human side of it. So here's what we found in our results. So just one slide briefly on our paper a couple years ago that was just about the 100 targeted chemicals. We found substantial differences in exposure by country. So this is across 6 different chemical classes. You just look very big picture here without getting into the weeds of it. You can see country on the x-axis with different chemicals colored differently for each class. You can see that the exposures vary a lot by country. And some of our key takeaways were that they were being exposed often to legacy chemicals like PCBs or old flame retardants or pesticides, even if they had been phased out decades ago. The office workers were also being exposed to chemicals that weren't banned in certain countries, unlike in other countries. So you can see that, for example, with some of the pesticides that aren't restricted in India like the other countries, which could be due to malaria control. And then finally, widely exposed to substitute chemicals used to replace older legacy chemicals, including some novel flame retardants and phthalate alternatives. So that was about the known chemicals, but now we wanted to know more about the potentially unknown chemicals. So in our suspect screening analysis, we observed about 1,000 chemical signatures across all the wristband samples. So each participant on average was exposed to roughly 800 different chemical features. Among the ones that we could tentatively identify as certain chemicals, the most reported source for those was fragrance, and about a third of those. And so now that's a lot of data with all these hundreds of chemical features. So now we wanted to get at how much together are these unknown chemical mixtures potentially disrupting hormones. What we found is that every single wristband sample was hormonally bioactive, so all 240. And I want to note that's unlike the field lengths that were mostly all below the limit of detection. So it wasn't the base material that was causing that. And the interpretation of this is that in human cells, the chemical mixtures that they were exposed to in office buildings could mimic or block hormones. 99% of the samples blocked thyroid hormone at its receptor, 96% blocked testosterone at its androgen receptor, and about 58% mimicked estrogen at its receptor. What I found was really interesting is what seemed to be a gender disparity, where the female office workers had exposures that were significantly more estrogenic, significantly more anti-androgenic, and more complex. More complex meaning they had significantly higher numbers of different chemicals in their wristbands. We think a large reason for this is that they were also reported to be heavier users of certain cosmetics like fragrance, perfume, and makeup and other products. I do want to note one disclaimer: we can't exclude the possibility that there could be natural substances on the skin that are picked up by the wristband, and that might differ by sex. To sort of control for this, one thing we did was adjust for the levels of sebum skin oil on the wristbands in our models. So the results still held within every country and even within shared office buildings. So that was really interesting. In terms of country effects, the workers in the UK tended to be exposed to less hormonally active mixtures, which could be due to their tighter restrictions on phthalates and certain pesticides, although we can't exactly know why that is. And then we also wanted to be able to look at a mixture effect and how all the chemicals together are influencing these hormonal activities. So this is statistically a challenge because there's, dozens of collinear chemicals. And the approach we chose is to use BKMR models as a way to get a total mixture effect, to get the single effects of individual chemicals within the mixture, while having flexibility with nonlinear and non-additive relationships. They also let you look at potential interactions between chemicals, although in our case, nothing was actually significant for the interactions. So what this showed us is that the higher the levels of all the measured chemicals in the samples, the higher the hormonal activities. So this was true for separate BKMR models we did for common targeted chemicals and common suspect chemicals. So you can see in these graphs here on the x-axis, you have increasing quantiles of joint chemical concentrations. So as the measured chemicals are simultaneously increasing, in our model, it's associated with strongly increasing hormonal activity outcomes for all three outcomes here. So we're seeing this strong mixture effect. And then we wanted to also be able to look at the single effects. So within those BKMR models, we identified several known and unknown chemicals that were important individual drivers of those overall mixture effects. So this is just one example graph of many for some common targeted chemicals and estrogen receptor activation. And you can see how certain chemicals pop out as major drivers. And what was interesting is that some of these drivers did have known sources, like as plasticizers, fragrance, sunscreen, or pesticide ingredients, but there were also other chemicals we could tentatively identify, but have unknown sources, or chemicals we couldn't identify at all. So there's still a lot unknown here. Beyond just the statistically significant chemicals, many others could have had contributing effects for the overall mixture effect. Because, for example, over 130 chemical signatures, these red dots here, were significantly and at least moderately correlated with each hormonal activity outcome. So there's, and that's after adjusting for multiple testing. So there are a lot of relationships going on here. And overall, this points to the fact that traditional targeted analyses alone are likely missing many hormonally active or unknown chemicals. So one last takeaway I thought was interesting is that so about 6 to 7 participants per office building were in our study, so we could look at how similar were people's exposures within the same office building. And we actually found that the vast majority of variability in hormonal activities was due to differences between individuals within the same building, not to differences between different buildings. So what this points to is the fact that activities and products can vary a lot within a building. Rooms and microenvironments can have different furnishings, dust loading, occupancy, and then as people are walking around and moving, they can kick up dust and create these activity clouds that move around with them. And then finally, we also did see that chemicals in personal care products and cosmetics and other products used at the individual level are important too. So overall, the takeaways were that their exposures were highly complex chemical mixtures, included many usually unknown or usually unmeasured chemicals, showed strong interference with hormone receptor function, demonstrated potential gender disparities, and were influenced by not only buildings, but also personal care products and other individual factors. And I think this also highlights that silicone wristbands can be a novel way to get at our personal exposures in the external environment around us and can capture these complex chemical mixtures. So I'd like to thank all of our collaborators on this, including my advisor, Joseph Allen, who's director of the Harvard Healthy Buildings program, our collaborators at Duke, Nick Herker and Heather Stapleton, others at Harvard, Brent Kuhl and Russ Hauser, Dr. Thomas Weller from UMass Amherst, as well as Peter Banish, Abraham Brower, and Emil Felzel from the Cell Assay Lab in the Netherlands. So I think Kristen will be able to share some links in the chat. This recent study, there's a special link you can get free full access until March 1st. And then we also have a separate paper from a couple years ago that was about the targeted chemicals. So I think we have time for questions now.
I want to know whether you, how you reported the results back to either the companies that were involved in helping you locate participants, or to the participants themselves.
Right. So, yep. So the participants were part of this larger global cog effect study focused on indoor air quality and cognitive function, actually. So they had a lot of devices that gave them real-time information, like their indoor air quality and Fitbits. For the wristbands, we didn't give individualized reports because the interpretation of chemical levels in wristbands are more challenging in terms of should I be concerned or not, since we couldn't like translate it to like looking at reference doses based on blood or urine levels. So we really just focused on summarized takeaways.
Okay. And how about to the companies? Did the companies respond at all to your findings in terms of how they might talk to their employees about avoiding certain products or anything like that?
Yeah, the companies seem very involved and engaged, which I think is why they participated in the research, so they could find out. And some of them have been communicating the results of the study too, which is great. And yeah, we have, so Harvard has partnered with some large organizations too to commit to only purchase healthier materials in their buildings for certain product categories. So there's a lot happening on that space too of pushing office buildings towards healthier materials.
That's really great that it has that kind of impact. How well does your findings correlate with what might be found in blood or serum or urine for some of the chemicals? I know that some of the chemicals have been tested in these other kinds of matrix. So what kind of, is there any kind of correlation between that? You probably get asked this question a lot, but I thought it'd be good for you to respond now to this.
Yeah, thanks for the questions. So a lot of work has been done by Heather Stapleton and Kim Anderson's group, correlating the wristband levels to blood or urine biomarkers, and they found good correlations with many types of chemicals. So that's reassuring that the wristbands are capturing something that we're actually exposed to. But they do sample a little differently in that it's only the external environment. It's not diet or water in that way. Yeah.
Okay, we have one question here in the question and answer. Is the silicon for the study different from what is used in food contact applications? Why are we baking and cooking with these substances if they're known to absorb chemicals so readily?
Yeah, that's a good question. So we buy these wristbands from this website online, 24hourwristbands.com. So they're just like those Livestrong bracelets that you see in the real world. I am not familiar with any research on testing the silicone baking materials. So I'd be curious to see that too. I know some people have talked about medical grade silicone should be less toxic, but yeah, I think I'm not as familiar with that research.
Oh, there is a scientist named Dr. I remember his name, Stewart, that actually studied silicone coated kitchen spoons that were used to stir soup, and found that these silicone utensils would absorb some of the chemicals in the kitchen, and then when they were used in soup, then these chemicals would show up in the soup. I'll see if I can find that and send it.
Really interesting.
Yeah, so there it is, folks. You might want to avoid silicone coated anything if you're going to eat from it. Okay, then RJ Herman, did you test the reaction to silicone alone?
Yes, so we did about 23 field blanks, which were silicone wristbands that hadn't been worn by anyone, and they were almost all below the limit of detection in the cell assays. So that was definitely very important to know, for us to know that it wasn't, the sampling device itself that was interfering with that.
Okay, and then from Sarah Howard, it seems like in India there were higher levels of most chemical exposures of known chemicals. but the hormone activity was similar to other countries. Is that to unidentified chemicals or something else?
Yeah, so the country differences in the hormone activity were interesting because we couldn't clearly link it to which chemicals specifically were higher in those countries. And I think that's because there's so many chemicals that differ a lot by country that there wasn't one obvious story there. But it's true. So India, for example, had higher exposures to pesticides and PAHs, but the flame retardants were actually higher in the US and UK, probably due to more historic strict fire codes for furniture and things like that. So yeah, so there were some exposures higher, but some lower. Yeah.
It's really interesting. Any studies in the future planned on the effect of ventilation on offer workers' exposure? Often green building initiatives focus on reducing energy use, but without any regulatory air quality standards. Those initiatives result in reducing energy use by reducing ventilation. So comment on that, yeah.
Yeah, that's a really great point. So yeah, actually the part of the larger parent study is has shown a lot on how ventilation in buildings and better indoor air quality improves our cognitive function, excuse me, cognitive function, performance, productivity. So that's been, so if you're interested in that, I can point you towards that research too. I think we could use some more studies on like building interventions and how they would reduce these like complex chemical mixtures. Because that would be a really actionable, simple way to target all those chemicals at once.
I know you're really dedicated to creating more healthy buildings. We're all really grateful for the work you do in that direction for sure. So someone anonymous, have you considered looking at certain more vulnerable populations in their indoor environments, workers in factories, construction, children in schools, residents in public housing? Have you thought about expanding your study to those kinds of populations?
Yes, we're definitely interested in that. I'd be curious more about like the cleaning workers in the buildings who are, in really direct contact with the dust and materials too, and seeing what their exposures are like. I also want to look more into disparities from the cosmetic chemicals. As we saw, there seemed to be a lot of contribution from cosmetics, which would be a disparity for women, and especially for women of color with certain hair products and other beauty products. So yeah, that's a really good point to mention.
Yeah, sure is. Okay. How concentrated are the extracts? An example of 20 milliliters wrist volume extracted to 20 UL. This is from Fred Schofele. And so I'm reading his question directly to you. Does this make sense to you?
Yeah, thank you for the question. So in the cell assays, they actually do 5 different dilutions of each wristband. So you can sort of get a dose response curve. So like seeing as that chemical mixture from that sample is getting more and more concentrated, how much more hormonal activity is happening. So that's what they're looking at. They're looking at a wide range of those extract dilutions.
That's really good. Okay, I think that we have come to the end of our time with you. Great. And we could ask questions for quite some time. But I think it's time for us to sign off. So I'm going to thank you for your great research. And I'm looking for more studies to come from your direction. And this is really important what you're doing, mixtures, untargeted analysis, chemicals, and so on. So I'm going to turn this webinar back over to Rachel to close us all out. And thanks a lot. And I look
Thank you so much
talking to you more.
again. Yeah, and thank you for the great questions.
You're welcome. Bye-bye.
Great. Thank you so much, Cheryl, and thank you very much, Dr. Young, for this really excellent presentation on this really important work. And thanks also for the thoughtful questions from today's participants. Let's see, so we already put a couple of additional links in the chat. So you look at those links, you can see the special link for the study that was discussed today, as well as the previous study on the targeted chemicals. And then just a few quick announcements as we wrap up. First, a video recording will be available on CHE's website soon, and everyone who registered for today's session will receive an email with a link to that video. Please keep an eye out for details on the next EDC Strategies Partnership call. You'll find details on the CHE website at healthandenvironment.org as soon as they're available. And we also invite you to join us next Thursday, February 23rd, for our next CHE webinar, Environmental Justice and the Built Environment, Protecting Children and Families. This will be the first of a three-part series on building products, children's health, and environmental justice, co-hosted with Blue Green Alliance, Healthy Building Network, and the Program on Reproductive Health and the Environment. And we'll put that link into the chat now. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on CHE's partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and or any of these partners. With that, I'd like to again thank Dr. Young for taking time to share this important research with us today, Cheryl for her excellent moderation, CHE Director Kristen Schaefer for behind-the-scenes support, and all of you for joining us today. Thank you so much.